Dear student, I am Dr. 𝗏𝐿𝐼, Professor of Biochemistry. In this lecture, I will be explaining you transcription and RNA processing. Let's get into the details. The first stage in the expression of genetic information is transcription of the information in the base sequence of a double-stranded DNA molecule to form the base sequence of a single-stranded molecule of RNA. For any particular gene, only one strand of the DNA molecule, that is the template strand, is copied by the DNA polymerase as it synthesizes RNA in the 5' to 3' direction. Because RNA polymerase moves in the 3' to 5' direction along the template strand of DNA, the RNA product is antiparallel and complementary to the template. RNA polymerase recognizes start signals, which are referred as promoters, and stop signals, which are referred as terminators, for each of the thousands of transcription units in the genome of an organism. So the figure below here illustrates the arrangement and direction of transcription for several genes on a DNA molecule. Now let's move on to see types of RNA. RNA molecules play a variety of roles in the cell. The major types of RNA are ribosomal RNA, referred as rRNA. This is the most abundant type of RNA in the cell. It is used as a structural component of the ribosome. The ribosomal RNA associates with the ribosomal proteins to form the complete functional ribosome. Now, the second type of RNA is a transfer RNA, which is referred as tRNA. This is the second most abundant type of RNA. Its function is to carry amino acids to the ribosome, where they will be linked together during protein synthesis. Now, coming to the third type of RNA, it is a messenger RNA, referred as mRNA. This carries the information specifying the amino acid sequence of a protein to the ribosome. Messenger RNA is the only type of RNA that is translated. The mRNA population in a cell is very heterogeneous in size and base sequence, as the cell has essentially a different mRNA molecule for each of the thousands of different proteins made by that cell. Coming to fourth type of RNA, that is heterogeneous nuclear RNA, referred as hnRNA, or also referred as pre-mRNA. This is found only in the nucleus of eukaryotic cells. It represents precursors of mRNA, and these are formed during its post-transcriptional processing. And then the last type of RNA is the small nuclear RNA, referred as snRNA. These are also found only in the nucleus of eukaryotes. One of its major function is to participate in splicing. Splicing means removal of introns, which are not coding for any proteins present over mRNA. So that's what is the role of small nuclear RNA. So they do the splicing and remove the introns and join the exons there. Now coming to the ribozymes. Ribozymes are RNA molecules with enzymatic activity, and they are found in both prokaryotes and eukaryotes. And these are the different types of RNAs. Now let's move on to see RNA polymerases. So the table here, it gives a difference between RNA polymerases that are found in prokaryotes and RNA polymerases that are found in eukaryotes. First, let me explain you or go over the points that are given for RNA polymerases found in prokaryotes. So, there is a single prokaryotic RNA polymerase that synthesizes all types of RNA in the cell. And the core polymerase responsible for making RNA polymerase as a subunit structure, that is alpha 2 beta beta prime. A protein factor called sigma is required for the initiation of transcription at a promoter. A sigma factor is released immediately after initiation is retake. A sigma factor is released immediately after initiation of transcription. Now, the termination of transcription sometimes requires a protein called Rho factor And the prokaryotic RNA polymerase is inhibited by rifampin, while actinomycin D will bind to DNA and prevent transcription process. Coming to RNA polymerases seen in eukaryotes. There are three eukaryotic RNA polymerases, as you can see here, which are distinguished by the particular types of RNA they produce. Now, the RNA polymerase 1 is located in the nucleolus and it synthesizes 28S, 18S and 5.8S RNAs. Whereas RNA polymerase 2 is located in the nucleoplasm and synthesizes heterogeneous nuclear RNA and mRNA and sometimes snRNA. RNA polymerase 3 is located in the nucleoplasm and synthesizes tRNA and some snRNA and 5S ribosomal RNA. Now, the transcription factors such as transcription factor 2D for RNA polymerase 2, it helps to initiate transcription process. The requirement for termination of transcription in eukaryotes are not well understood. All transcription can be inhibited by actinomycin D. In addition, RNA polymerase 2 is inhibited by alpha amanitin, which is a toxin from certain mushrooms, that is Amanita phyllaides mushroom. Now let's move on to see transcription process. RNA is synthesized by DNA-dependent RNA polymerase, which uses DNA as a template for synthesis of RNA. Now, RNA polymerases locates genes in DNA by searching for a promoter regions. The promoter is the binding site for RNA polymerase. Binding establishes where transcription has to begin, which strand of DNA is used as the template, and in which direction transcription proceeds. And no primer is required. And the RNA polymerase moves along the template strand in 3' to 5' direction as it synthesizes the RNA product in the 5' to 3' direction using nucleotide triphosphates, that is ATP, GTP, CTP and UTP as the substrates. RNA polymerase does not proofread its work. The RNA product is complementary and anti-parallel to the template strand. The coding or the antiplatelet strand is not used during transcription. It is identical in sequence to the RNA molecule, except that RNA contains uracil instead of the thymine found in DNA. By convention, the base sequence of a gene is given from the coding strand and 5' to 3' direction. In the vicinity of a gene, a numbering system is used to identify the location of important bases. The first base transcribed as RNA is defined as the +1 base of that gene region, as it is shown in the figure here. To the left, that is a 5' or the upstream of this start point for the transcription, bases are written as -1, -2, -3 like that. To the right, that is 3' direction or the downstream of this point, bases are usually mentioned as +2, +3, etc. Transcription ends when RNA polymerase reaches a termination signal. So that's the brief introduction to transcription process. Now the flow of genetic information will happen from DNA to protein. So the messenger RNA, which is synthesized in 5' to 3' direction, it is complementary and anti-parallel to the template strand of DNA. The ribosome translates the messenger RNA in 5' to 3' direction, and a protein is synthesized from the amino terminus to the carboxy terminus. And this is how the directionality will go on. For the case of gene coding for a protein, the relationship among the sequence found in a double-stranded DNA, a single-stranded mRNA and protein is illustrated here in this figure. Messenger RNA is synthesized in the 5' to 3' direction. It is complementary and anti-parallel to the template strand of DNA. The ribosome translates the mRNA in the 5' to 3' direction as it synthesizes the protein from the amino terminus to the carboxy terminus. Now let me explain you a process of transcription of prokaryotic messenger RNA. The structure and expression of a typical prokaryotic gene coding for a protein is given in this particular figure. Let me explain the process. With the help of a sigma factor, RNA polymerase recognizes and binds to the promoter region. The bacterial promoter contains two consensus sequences called the prin box or a Tata box and the -35 sequence. The promoter identifies the start site for transcription and orients the enzyme on the template strand. And then the RNA polymerase separates the two strands of DNA as it reads the base sequence of the template strand. The transcription begins at the +1 base pair. A sigma factor is released as soon as transcription is initiated. And the core polymerase continues moving along the template strand in 3' to 5' direction, synthesizing mRNA in the 5' to 3' direction. Now then RNA polymerase eventually reaches a transcription termination signal, at which point it will stop transcription and release the completed mRNA molecule. So there are two kinds of transcription terminators commonly found in prokaryotic genes. One is the row independent termination, which occurs when the newly formed RNA folds back in itself to form a GC-rich hairpin loop closely followed by 6 to 8 uracil residues. These two structural features of newly synthesized RNA promote dissociation of the RNA from the DNA template. This is the type of termination that is shown here in this particular figure. Row-dependent termination requires participation of row factor. This protein binds to the newly formed RNA and moves toward the RNA polymerase that has paused at the termination site. And the row then displaces RNA polymerase from the 3' end of the RNA. Now, the transcription and translation can occur simultaneously in bacteria. Because there is no processing of prokaryotic mRNA, meaning there is no introns, ribosomes can begin translating the message even before transcription is complete. Ribosomes bind to a sequence called 𝐮𝐬𝐜𝐫𝐢𝐛𝐞 sequence in the 5' untranslated region of the message, as it is shown here in the figure. And protein synthesis begins at an AUG codon at the beginning of the coding region and continues until the ribosome reaches a stop codon at the end of the coding region. The ribosome translates the message in the 5' to 3' direction, synthesizing the protein from the amino terminus to the carboxy terminus. So all the points that I have explained in the previous slide, so I have written here. So let me just quickly go over those points as a recap. Sigma factor, RNA polymerases, promoter region, prin box and 35 sequences. These are some of the things that are found where the DNA is undergoing a transcription process. Transcription, it begins at the +1 base pair. RNA polymerase transcribes the template strand in 3' to 5' direction, synthesizing mRNA in the 5' to 3' direction. Now, at the transcription termination signal, RNA polymerase stops transcription. There are two kinds of transcription terminators. One is row independent termination and the other is row dependent termination. Transcription and translation can occur simultaneously in bacteria because there are no introns in the bacterial DNA. Now, the ribosome translates the messenger RNA in 5' to 3' direction. The protein is synthesized from amino terminus to the carboxy terminus. And the monocistronic message, where a single gene is transcribed to an mRNA to make a single protein, is done. Now, the mRNA produced by the gene, as we have explained in the previous figure. It is the monocistronic message, that is, it is transcribed from a single gene and codes only for a single protein. The word cistron here is another name for a gene. So, as you are seeing here in this particular figure, in this slide, some bacterial operons, like example lactose operon, they produce polycistronic message. In these cases, related genes are grouped together in the DNA and are transcribed as one unit. And the mRNA in this case contains information from several genes and codes for several different proteins. That's how a polycistronic mRNA is transcribed to give more than one protein molecule. Now let me explain you transcription of eukaryotic messenger RNA. In eukaryotes, most genes are composed of coding segments referred as exons, and these exons are interrupted by a non-coding segments, introns. Both exons and introns are transcribed in the nucleus. Introns are removed during processing of the RNA molecule in the nucleus. In eukaryotes, all mRNA is a monocistronic mRNA. And the mature mRNA is translated in the cytoplasm. Now let me explain you how the transcription goes on in a eukaryotic cells giving rise to messenger RNA. With the help of proteins called transcription factors, RNA polymerase 2 recognizes and binds to the promoter region. The basal promoter region of eukaryotic genes usually has two consensus sequences called Tata box, sometimes referred as Hogness box, and the cat box. RNA polymerase 2, it separates the strands of the DNA over a short region to initiate a transcription and read the DNA sequence. The template strand is read in the 3' to 5' direction as the RNA product, that is the primary transcript, is synthesized in the 5' to 3' direction. Both exons and introns are transcribed. And then the RNA polymerase 2 ends transcription when it reaches a termination signal. And these signals are not well understood in eukaryotes. So, the summary here is, in eukaryotes, most genes are composed of coding segments, that is exons, and interrupted by non-coding segments, introns. Both exons and introns are transcribed in the nucleus. Introns are removed during the processing of the RNA molecule in the nucleus, and in eukaryotes, all mRNA is monocistronic and mature mRNA is translated in the cytoplasm. RNA polymerase 2, with the help of transcription factor, recognizes and binds the promoter region, the Tata box, and the cat box. RNA polymerase 2 separates DNA strands and read the template strand in 3' to 5' direction, and the transcription ends when RNA polymerase reaches yet poorly understood termination signal. Alright, now let me explain you processing of eukaryotic pre-messenger RNA. The primary transcript coming out of eukaryotic gene transcription process should undergo extensive post-transcriptional processing inside the nucleus to form the mature mRNA molecule. So here in this post-transcriptional processing, a 7-methyl-guanosine cap is added to the 5' end while the RNA is still being synthesized, as it is shown here in the figure. The cap structure serves as a ribosome binding site, and also it helps to protect mRNA chain from degradation. And then a poly-A tail is attached to the 3' end. In this process, an endonuclease cuts the molecule on the 3' side of the sequence, which is 𝐴𝐴-𝐸𝐴𝐴, that's referred as poly-A addition signal. Then the poly-A polymerase adds the poly-A tail. About 200 adenines are added to the 3' new end. The new poly A tail protects the message against rapid degradation and aids in its transport to the cytoplasm. A few mRNAs, for example histone mRNAs, have no poly A tails. So introns are removed from hnRNA by splicing, accomplished by spliceosomes, which are also known as snRNAs or SNRPs, which are complexes of small nuclear RNAs and proteins. The hnRNA molecule is cut at splice sites at the 5' donor and 3' acceptor ends of the introns. The intron is excised in the form of a lariat structure and it will be degraded. And the neighboring exons are joined together to assemble the coding region of the mature mRNA. And this is how all of the intermediates in this processing pathway are collectively known as hnRNA. The mature mRNA molecule is transported to the cytoplasm where it is translated to form a protein molecule. So let's recap the processing of eukaryotic pre-messenger RNA. Steps in pre-mRNA conversion to mature mRNA is addition of 7-methyl-guanosine cap at the 5' end, addition of poly A tail at the 3' end, and the poly A addition signal sequence is AAUAAA. And the spliceosome removes introns from hnRNA, which is referred as splicing. All exons are joined to make a mature mRNA, and the mature mRNA in the cytoplasm is translated to form a protein. So this is in brief and how a pre-mRNA is processed into mature mRNA. Now let's move on to see something called as alternate splicing that is done in eukaryotic pre-mRNA molecule. For some genes, the primary transcript is spliced differently to produce 2 or more variants of a protein from the same gene. This process is known as alternate splicing. The variants of the muscle proteins like tropomyosin and troponin T are produced in this way. The synthesis of membrane-bound immunoglobulins by unstimulated B lymphocytes as opposed to the secreted immunoglobulins by antigen-stimulated B lymphocytes are also involves alternate splicing. So, the primary transcript from a large percentage of genes undergoes alternate splicing. This may occur within the cell or the primary transcript of a gene may be alternatively spliced in different tissues, giving rise to a tissue-specific protein products. And by alternate splicing, an organism can make many more different proteins than it has genes to encode for. A current estimate of the number of human proteins is at least 100,000, whereas the current estimate of human genes is only about 20 to 25,000. And note that alternate splicing can be detected by a northern blot technique. And the alternate splicing mechanism is briefly shown here in the figure. Now let me explain you ribosomal RNA, which is referred as rRNA. The figure here, it shows a ribosomal RNAs found in prokaryotes and also eukaryotes. Eukaryotic ribosomal RNA is transcribed in the nucleolus by RNA polymerase 1 as a single piece 45S ribosomal RNA, which is subsequently cleaved to yield 28S ribosomal RNA, 18S ribosomal RNA and 5.8S ribosomal RNA. And the RNA polymerase 3 transcribes the 5S ribosomal RNA unit from a separate gene, and that's how the eukaryotic ribosome is made. So the ribosomal subunit assemble in the nucleolus as the ribosomal RNA pieces combine with the ribosomal proteins. And the eukaryotic ribosomal subunits are 60S subunit and 40S subunit, and they join during the protein synthesis to form a whole 80S ribosome, and that's how the 80S ribosome is made. The large and small prokaryotic ribosomal subunits, that is 50S subunit and 38 subunits respectively. The complete prokaryotic ribosome is a 70S particle. The S value here determined by the behavior of the particles in an ultracentrifuge. They are a function of both the size and shape, and therefore the numbers are not additive. We should not add the numbers there. So that's how prokaryotic ribosome and eukaryotic ribosomes are seen. So just to recap what we have seen in the previous slide, RNA polymerase 1 transcribes eukaryotic ribosomal RNA in the nucleolus to form 45S RNA, which is cleaved into 28S rRNA, 18S rRNA and 5.8S rRNA. RNA polymerase 3 transcribes the 5S ribosomal RNA separately, and eukaryotic ribosomal subunits are 60S and 40S, and they both combine during protein synthesis to make an 80S ribosome. The prokaryotic ribosomal subunits are 50S and 30S. They both combine during protein synthesis to make 70S ribosome. Note that in prokaryotic 70S ribosome, so the 50S subunit contains 5S rRNA and 23S rRNA, whereas 30S subunit contains 16S rRNA. Now let me explain you transfer RNA. This transfer RNA which carries activated amino acids for translation process. There are many different specific tRNAs or transfer RNAs, and each tRNA carries only one type of activated amino acid for making proteins during translation. The genes encoding these tRNAs in eukaryotic cells are transcribed by RNA polymerase 3 enzyme. The tRNAs enter the cytoplasm where they combine with their appropriate amino acids. And although all tRNAs have the same general shape, but small structural features will distinguish among them. So here is the structure for tRNA. It has got a 5' end which is phosphorylated, and then it has got a 3' end which is having a hydroxyl group, where the activated amino acid is attached to this 3' hydroxyl group. And also it has got an anticodon sequence, that is CAU pair with the codon in mRNA. Basically, the anticodon sequence present in tRNA, it will be specific for each amino acid that it is carrying, and this anticodon is going to pair with the codon present over mRNA. That's how it is bringing the amino acids and pairing with the codons in mRNA, and thereby it is helping in building a polypeptide chain. So there are many different specific tRNAs for carrying one specific activated amino acid for making proteins during translation. RNA polymerase 3 transcribes tRNA from specific genes, and tRNA in the cytoplasm combine with specific amino acid for translation. Now let me explain you what is RNA editing process. RNA editing is a process by which some cells make discrete changes to specific nucleotide sequences within the RNA molecule after its gene has been transcribed by RNA polymerase. The post-transcription editing events may include insertion, deletion, or base alterations of nucleotides, such as adenine deamination. So this happens within the edited RNA molecules. RNA editing has been observed in some messenger RNA, ribosomal RNA, transfer RNA molecules in humans. An important example in cytosine to uracil deamination process can be explained by using mRNA editing that happens over the messenger RNA that come from ApoB gene. So apoprotein B100 is expressed in the liver, and apoprotein B48 is expressed in the intestine. So in the intestine, what happens is the mRNA which is coming from the expression of apolipoprotein B100 gene is edited, the CAA which is coding for glutamine, and the cytidine in that CAA codon will undergo deamination process to make uracil. So CAA becomes UAA because of action of cytidine deaminase, and UAA is a stop codon, and that means by 48% of the mRNA that is being translated, you get that apoprotein B48 instead of apoprotein B100. So, apoprotein B100 gene, which is actually coding for ApoB100, 100% of the protein, it is now only 48% of it is translated because of appearance of stop codon at that 48%. That happens because of CAA, which is converted into UAA. So this is one of the example for RNA editing, that is mRNA editing in this particular example. So that's about transcription and RNA processing. Thank you.